Okay, Bismillah Rahman Rahim. The topic of our today's lecture is importance of secondary metabolites in organisms, in microorganisms. So you know in the previous lecture that metabolites are basically of two types. One are the primary metabolites, and the second ones are the secondary metabolites. So primary metabolites, as we have already discussed in the previous lecture, they are thought to have a great importance in the growth as well as survival of the microorganisms. While the secondary metabolites are thought that they do not have any apparent role in the growth and survival of the microorganisms. Then question is why these secondary metabolites are released extracellularly by the microorganisms. Why these secondary metabolites are produced and then they are secreted extracellularly by the microorganisms. So in order to explain the importance of secondary metabolites in microorganisms, there are some theories and hypotheses. So today we are going to discuss those theories and hypotheses that tries to explain the importance of secondary metabolites in microorganisms. So there are some old theories, there are some earlier views about the importance of secondary metabolites. Number one is the food storage materials. So this theory explains that uh, the secondary metabolites are actually the food storage materials. And the second one states that the secondary metabolites are waste products actually. So they are the waste products of metabolism and that originate by the breakdown of larger molecules. So these are some of the earlier thoughts about the secondary metabolites. Now we are going to discuss some current hypotheses that exist in order to explain the importance of secondary metabolites. So the first hypothesis about the importance of secondary metabolites is the competition hypothesis. So competition hypothesis states that the secondary metabolites are produced due to the competition of the microorganisms in order to get food in a particular ecosystem. So this means whenever the microorganisms they grow in a particular ecosystem and they tries to get food and they compete with other microorganisms in order to get that food. So they compete, and in this competition they secrete many antibiotics for an effective competition. So this hypothesis states that in order to support this hypothesis, there are some evidences, and one evidence is the distribution of wide distribution of beta-lactamases enzymes in the microorganisms. So presence of these beta-lactamases in microorganisms explains the competition hypothesis. Due to these beta-lactamases, microorganisms have the ability to detoxify many beta-lactam antibiotics. So, you know, beta-lactam antibiotics are a group of antibiotics, and one example is a very famous one, penicillin. So, in order to detoxify those antibiotics in the environment, they have a wide distribution of beta-lactamases. So, this hypothesis, the distribution of these beta-lactamases supports this hypothesis of competition, that these features enable microorganisms in order to better compete in the environment. And but there are some limitations exist of this hypothesis, and the limitations are the restriction. This hypothesis is restricted to antibiotics only, and you know that there are a variety of secondary metabolites that are being produced by the microorganisms, and this secondary metabolism is not only limited to antibiotics. There are a variety of other compounds also exist in secondary metabolites. So but this hypothesis, the explanation of this hypothesis is only restricted to the antibiotics. This is a limitation. And the second limitation is, even in case of antibiotics, even if you consider the antibiotics, there are some antibiotics, or in fact, there are many of the antibiotics that are actually outside the beta-lactam spectrum. So there are many antibiotics that exist outside beta-lactam category. So this hypothesis does not explain anything about those antibiotics as well. Then the second hypothesis about the importance of secondary metabolites in the microorganisms is named as the maintenance hypothesis. So the maintenance hypothesis states that the secondary metabolites are also responsible for the growth as well as the maintenance of the microorganisms. And this is exactly like the primary metabolism. Primary metabolites also, the concept of primary metabolites also states that these are those metabolites that are directly responsible for the growth and survival of the microorganisms. So maintenance hypothesis tries to explain the necessity of the secondary metabolites towards the maintenance and towards the growth and towards the cell division of the microorganisms. So why this hypothesis is of the view that secondary metabolites are directly linked to the growth and cell division? This is because this hypothesis states that secondary metabolites are always produced whenever a vital nutrient is exhausted in the media. For example, the carbon sources, for example, the glucose, the lactose, these are the vital nutrients. So whenever the vital nutrients are exhausted in the media, then there comes the production of the secondary metabolites in the media. So this means that the enzymes that are responsible for the primary metabolism in the microorganisms, whenever the nutrient is exhausted, these enzymes are no longer required. So when these enzymes are no longer required, they are destroyed to produce the precursors of the secondary metabolites. So this is the complete linkage of the maintenance hypothesis. So to summarize that, this hypothesis states that the secondary metabolites are also linked towards the growth and cell division, because this hypothesis states that whenever a vital nutrient is exhausted in the media, then this means that the primary metabolites, primary enzymes that are responsible for the primary metabolism, they are no longer required, and then these are destroyed to the precursors of secondary metabolites. But there is a limitation of the maintenance hypothesis as well. So the limitation of maintenance hypothesis is that microorganisms can still grow, they can still divide without entering even into the secondary metabolism. So if the microorganisms do not enter into the secondary metabolism, even then they are able to grow, even then they are able to survive in the environment. So this is a limitation. Then there is a third hypothesis about the importance of secondary metabolites in the microorganism. The third hypothesis is the unbalanced growth hypothesis. So unbalanced growth hypothesis is very simple and straightforward. This hypothesis states that the primary metabolites are no longer, there is no ability of the microorganisms to store the primary metabolites. And the control mechanism in the microorganisms is much more weaker as compared to the higher organisms. So they are not able to control the primary metabolites once they are produced in huge numbers. So what happens that once the primary metabolites are produced in large amounts in the cells of the microorganisms, so what happens, these primary metabolites are then destroyed, they are converted into the secondary metabolites, because microorganisms is not able to store these primary metabolites. And by destroying these metabolites into the secondary metabolites, then these secondary metabolites are secreted out of the cells of the microorganisms. So this is the unbalanced growth. If there is no secondary metabolism, if there is no synthesis of the secondary metabolites in the cells of the microorganisms, then the microorganisms would not be able to store, microorganisms would not be able to survive in the presence of huge amount of these primary metabolites. So this is all about the unbalanced growth hypothesis that states that If in the absence of the secondary metabolites, there might be the chances of the unbalanced growth in the microbial cells. Then there is a fourth hypothesis about the secondary metabolites, about the importance of secondary metabolites in microorganisms. And the fourth hypothesis states that the name of this hypothesis is the detoxification hypothesis. So this hypothesis is simply states that some of the primary metabolites or some of the molecules that are produced as a result of the primary metabolites, as a result of the primary metabolism in the microbial cells, they are so toxic for the microbial cells that this becomes a necessity for the microbial cells in order to convert them into the less toxic molecules. So this is detoxification hypothesis. Detoxification hypothesis simply is the secondary metabolism occur in microorganism uh, as a need of detoxification in the microbial cells. In the absence, if there is no secondary metabolism in the microbial cells, then microbial cells would be toxified. There are too many toxins that could arise in the microbial cell as a result of the primary metabolism. So, in order to explain this hypothesis, in order to explain the detoxification hypothesis in the microbial cells, there is an example, and the example is penicillinic acid. Penicillinic acid is actually a precursor of penicillin antibiotic. So whenever there is a penicillinic acid in the microbial cells, this penicillinic acid is more toxic as compared to original penicillin. So microbial cell try to convert this penicillinic acid into the penicillin uh, in order to keep the microbial cells safer, in order to keep the microbial cell toxin free. So this hypothesis states that there are so many toxins, there are so many toxic molecules in the microbial cells that are ultimately converted to less toxic molecules, and that those less toxic molecules would be then called as the secondary metabolites, and they are then secreted out of the cells. But this hypothesis again has some limitations. Again, this hypothesis is linked. This hypothesis explain the metabolism of antibiotics only. So this is only limited to antibiotic synthesis. So there are as I have already discussed that there are so many secondary metabolites present in microbial cells that are not antibiotics. In fact, variety of secondary metabolites are being produced by the microbial cells. So uh, only some precursors of the antibiotics are more toxic than original antibiotics as secondary metabolites. So this is one limitation. Another limitation of this hypothesis is that many precursors, yes, this point I have already explained, that many precursors, even in antibiotics, even if you talk about the antibiotics, many precursors of the antibiotics are even not toxic than the original antibiotics. So these are some of the limitations that this detoxification hypothesis has. Then there comes another hypothesis, which is the regulatory hypothesis. And regulatory hypothesis explains that the secondary metabolism is linked towards certain morphological changes. So this hypothesis is very important, and this simply states that whenever microbial cells produce secondary metabolites, they are undergoing some morphological changes in their cells as well. So in order to explain these morphological changes, in order to explain this linkage between the morphological changes and the synthesis of the secondary metabolites in the microbial cells, there are some examples. So one example is the production of antibiotics in Bacillus species whenever the Bacillus species are forming the spores. So when there is spore formation, when there is spore formation in the Bacillus species, there would always be the stage of antibiotic production. So this means that the spore formulation or the spore formation is directly linked to the antibiotic production in Bacillus species. Another example is the carotenoid synthesis in Neurospora crassa. This is a fungus, Neurospora crassa. So the carotenoid synthesis in Neurospora crassa always happen whenever there is a stage of sporulation. So this means that these morphological changes are directly linked towards the synthesis of secondary metabolites. So uh, another evidence, in order to support this regulatory hypothesis states that whenever we produce mutants of Bacillus species, uh, whenever we produce the mutants of the Bacillus species which do not have the ability of spore formation, what happens that they would not ultimately produce the antibiotics. So this evidence in many studies support this hypothesis actually, that yes, there are certain morphological changes that ultimately are linked towards the initiation of secondary metabolism. Then if if this happens that certain morphological changes are linked towards the secondary metabolism, then this directly means that secondary metabolism is also linked towards the external environment, right? So this means that whenever there are some external triggers, for example, temperature changes, for example, the changes in the sunlight, for example, there are the healthy environment or the harsh environment. So whenever there are some changes in the external environment, then this means that these external changes would directly are directly linked to the secondary metabolism in the microorganisms. So this is all about the regulatory hypothesis of the secondary metabolites. Then there is another hypothesis of secondary metabolites, and that is the evolutionary hypothesis. And evolutionary hypothesis is very important hypothesis that directly states that the synthesis of secondary metabolites in microorganisms is directly linked towards the evolutionary process in microorganisms. So, you know, in evolutionary process, the fundamental thing is only those traits are kept in microorganisms that are required with the evolutionary process, and all others are disappeared. This is the fundamental concept of the evolution. So this hypothesis states that some of the primary metabolites or some of the enzymes of the primary metabolites with the evolutionary process or with the passage of times are no longer required by the microorganisms. So they are then extra enzymes. So those metabolites or those products of primary metabolism which with the evolutionary process, with the passage of time are no longer required by the microbial cells, they are then broken down into the simpler components and those are the secondary metabolites. So in this process, in this way, these metabolites are ultimately released out of the cells. So this is the evolutionary process. So this is the evolutionary hypothesis. So if the evolutionary hypothesis is really a true happening about this synthesis of secondary metabolites in microorganisms, then this implies that our detection techniques are very limited in order to determine the secondary metabolites. So, for example, if this is true that the secondary metabolites are linked towards the evolution, then this means that evolution is a continuous process, right? Evolution is always undergoing in microorganisms, in other animals, in plants, and in all organisms. So evolution is a continuously growing process. So if this hypothesis is true, then this means that secondary metabolites are much more in number, are much more in variety as compared to whatever information we currently have, right? So this means that our detection techniques are so limited. Our detection methods are so simple, so limited that we are currently not able to determine or measure all the secondary metabolites that are being produced with the evolutionary process of the microorganisms. So this means that in future, if we are able to develop more sophisticated detection techniques in order to measure the secondary metabolites, then we would be able to determine more number and more variety of secondary metabolites of the microorganism. So I thank you all for today's lecture. This is all about today's lecture. Now, if you have any question,